Say everything is good with your cell and you want to get from the G1 phase into the S phase. How are you going to do that? Well, we have this transcription factor, E2F, which is referred to as an accelerator because what it does is help get through that restriction point. So it binds to the promoter region, which is before the S cyclin gene. It also acts on the G1S cyclin genes, and then those genes become the S cyclin or G1S cyclin mRNA, and then the corresponding actual cyclin protein, which remember is important because the G1S CDK and the S CDK need their corresponding cyclins to be able to work, and we need them to be able to go on into the S phase and replicate DNA. So if you want to get from G1 to S, well, you need to have these E2F transcription factors working and increasing the SCDK and G1S CDK activity. So if E2F is a transcription factor that's an accelerator and that it increases CDK activity, we can't just have an accelerator inside our body. We also need brakes because we're always trying to maintain a balance, right? So the brake for this accelerator, and it's always a partner that you want to have both of them to be able to control the cell cycle properly, we have the RB protein. So what it does is wrap around the E2F, and when the RB protein is active, the E2F is inactive and therefore can't initiate transcription of S cyclin or G1S cyclin, which means these types of cyclin will not be made, and then you won't be able to activate G1S and S CDKs. So the RB protein usually binds to the E2F during the G1 phase because you don't want it to go into the S phase at that time. But once you want to pass that restriction point and go from G1 into S, you need to get rid of this RB protein. How are you going to do that? Recall that the G1S restriction point partially depends on the environment that the cell is in. So if there's enough nutrients, if there's enough room and stuff for that, so that it can replicate and grow in that environment. So it makes sense that it would need a signal from that environment. In this case, the signal is mitogen. So mitogen binds to its receptor, and then a whole pathway goes along here, and then eventually it activates G1 CDK or G1S CDK. And we talked about those before, and they're important for getting through this particular checkpoint. So how they do that is by phosphorylating the RB protein. So once it is phosphorylated, it becomes inactive and releases E2F, which is now active. Once E2F is active, it can go do its job, transcription factor, and then initiate transcription of S cyclin or G1S cyclin, which are both important to be then later transcribed and translated into their corresponding actual cyclin proteins, activate their corresponding CDKs, and then the cell can go into S phase and continue to replicate.